Hello and welcome to the webinar. RNA quadruplex rG4 is self-assembled guanidine quartet structure in RNAs. rG4s are dynamically formed as novel RNA elements to regulate protein translation, RNA stability, and epitranscriptomic modifications. rG4s are associated with cancers and diseases, making them an important RNA feature for scientific and clinical research. RNA quadruplex is single-stranded RNA sequence with guanine repeats that can self-assemble into G-quartet through Hoogsteen hydrogen bonds. G-quartets are stacked together to form G-quadruplex. The sequence between the G-quartet tracks is called a loop. G-quadruplexes are stabilized by monovalent cations such as potassium or sodium ions, with thermal stability of potassium greater than sodium ion. RNA quadruplexes, rG4, are observed enriched in 5' or 3' UTR in mRNAs. They are transiently and dynamically formed, regulated by binding proteins, cellular stress, and environment. There are about 13,000 rG4 regions known. In solution, rG4 structure is thermally very stable in the presence of potassium ion, with melting temperature higher than 90 degrees. rG4 can be destabilized by small molecular ligands, divalent cations, for example calcium ions, helicases and EIF4A, and melting temperature. Conversely, rG4 can be refolded by monocations, for example potassium ions, stabilizing small molecule ligands, and RNA-binding proteins, RBPs. In mRNAs, rG4s are enriched on the 5' and 3' untranslated regions, suggesting their regulatory roles in protein translation initiation, RNA stability, and metabolism. In 5' UTR, rG4 can block ribosome scanning and impede canonical cap-dependent protein translation initiation. For cap-independent protein translation, rG4 in the internal ribosome entry site IRES can recruit translation initiation factors to start protein translation. This bifurcation in function underscores the versatility of rG4s. That is, cells can switch from cap-dependent to IRES-mediated translation mechanism, where rG4s is the key to this translational reprogramming. In the open reading frame, rG4s can be modulators of the translation elongation process. rG4s can also act as a barrier that slows down or temporarily pauses the ribosome during peptide synthesis. In 3' UTR, rG4s can mask the binding sites of microRNAs and block microRNA silencing. By forming or dissolving, rG4s can change the accessibility of miRNA target sites, thereby modulating the rate at which an mRNA is degraded or sequestered from translation. In 3' UTR, and in fact also in 5' UTR and CDS, rG4s can interact with various protein factors to do various functions. rG4s have many examples of diverse molecular functions in different host RNA types. In the nucleus, rG4s regulate RNA transcription, splicing, nuclear to cytoplasmic export. rG4 in long non-coding RNA tera regulates telomerase activity and telomere length. In the cytoplasm, rG4s can interact with or sequester RNA-binding proteins, RBPs, that play diverse functions and roles. rG4s in piRNAs can inhibit miRNA processing and maturation. In 3' UTR of mRNA, rG4s can affect miRNA targeting and mRNA silencing. In mRNAs, rG4s regulate RNA epitranscriptomic modification, mRNA translation, and RNA decay. In the cytoplasm, liquid phase separation forms membraneless organelles, signaling complexes, and many other biological supramolecular assemblies. rG4s trigger the phase separation. In mitochondria, rG4s can affect mitochondrial DNA replication, transcription, and mitochondrial RNA decay, and overall mitochondrial function. Dysregulation of rG4 can lead to mitochondrial dysfunction. If we look at rG4 in mRNA closely, EIF4A in the translation initiation complex can bind and resolve the rG4 during the translation initiation scanning process. The disrupted rG4 structure helps to form a molecular platform for recruiting the 40S ribosomal subunit. Key mRNA exemplars like BCL2, RUNX1, and CMYC incorporate rG4 structures as integral components in their 5' UTR, suggesting that the involvement of rG4 in translation initiation, and particularly its resolution at translation initiation, is not an isolated phenomenon but a common regulatory strategy for mRNAs encoding proteins in the development of cancers such as T-cell acute lymphoblastic leukemia. Under normal conditions, mRNA translation is largely driven by the recognition of the 5' cap by specific initiation factors. However, during stress or in certain pathological conditions, the cap-dependent machinery may be compromised and cap-dependent translation is stopped. However, mRNAs harboring internal ribosome entry site (IRES) can still be translated. rG4 within the IRES region directly facilitates the recruitment of the ribosomal machinery. By directly engaging with the translation initiation complex, rG4 help to form a molecular platform that recruits the 40S ribosomal subunit. The stabilizing role of rG4, which provide an alternative platform that mediates the assembly of translational complexes even when canonical translation mechanisms fail. In doing so, rG4 not only safeguard the synthesis of crucial proteins under adverse conditions, but also allow for a finely tuned regulation of protein output depending on the cell's needs. Here, FGF2 and alpha-synuclein mRNAs both serve as exemplars of incorporating rG4 as an integral component of the IRES in 5' UTR. URFs is a short upstream ORF located in the 5' UTR, whereas MORF is the main ORF for encoding the protein. URFs are mostly regulatory on the downstream main MORF translation, for example ribosome stalling or dissociation before reaching the main ORF, or trigger nonsense-mediated mRNA decay. rG4 in the 5' UTR can toggle the translation efficiency between UORF and MORF. In the absence of a stable rG4, the MORF tends to be translated. Conversely, with a stable rG4 in the 5' UTR, it can activate the translation of the UORF, which in turn suppresses the translation of the MORF. Thus, the rG4 acts as a molecular switch to fine-tune the selection of UORF and MORF during protein synthesis, thereby regulating the level of translation without the cost of changing mRNA expression level. DHX36 or DHX9. A helicase enzyme that can resolve rG4 plays a decisive role in determining whether the rG4 remains intact or is unwound, thereby determining whether the UORF or the MORF is translated. Thus, rG4 is not just a passive structural feature but an active regulator of protein synthesis. The rG4 dynamic process can determine the mRNA translation output. rG4 structures in ORF can serve as a blockade to ribosomal progression along the mRNA, effectively inhibiting the translation process. rG4 in the ORF of EBNA1 Epstein-Barr viral mRNA is a particularly striking example of rG4 regulation. EBNA1 is an essential viral antigen with its level needed to be critically balanced. On the one hand, it is crucial for the maintenance of the EBV genome within infected cells. On the other hand, its expression must be kept at relatively low levels to help the virus evade T-cell immune surveillance. So the EBNA1 protein level needs to be delicately balanced between sufficient viral protein production for viral persistence and minimal expression to avoid immune detection. The rG4 in the ORF of EBNA1 mRNA act to suppress the translation of the EBNA1 protein. This autoregulatory mechanism for the virus to self-limit the expression of EBNA1 protein prevents overproduction of the antigen, thereby reducing the likelihood of host immune response. The delicate balance can be broken for therapeutic applications. By using antisense oligonucleotides, ASO, targeting the rG4, the translational block can be relieved to enhance antigen presentation and antiviral immune response. Conversely, small molecule ligands such as PDS can stabilize the rG force to further suppresses EBNA1 translation and the virus. In a broader sense beyond EBNA1 viral RNA, rG force in other mRNAs may as well act as a molecular rheostat controlling protein synthesis. In fact, rG force are found in mammalian mRNAs such as MLL1 and MLL4, known to be involved in chromatin regulation and in leukemias. Here, rG force in the open reading frames of mixed lineage leukemia MM1 and MLL4 mRNAs are recognized by AVIN protein. AVIN recruits helicase DHX36 to unwind the rG4 in the ORF and facilitates a more efficient translation of MLL1 and MLL4 proteins. In 3' UTR, rG force can interfere with microRNA binding and affect mRNA stability. For example, the rG4 in the 3' UTR of FADS2 mRNA can physically mask the miRNA miR331-3P target site, prevent the microRNA binding and silencing, and positively upregulate FADS2 expression. As a result, the miRNA-mediated repression of the mRNA is diminished, thereby altering the balance between mRNA degradation and translation. This mechanism of action establishes rG4 structures as key modulators of mRNA stability and turnover, with significant implications for gene expression regulation. Additionally, rG force in 3' UTR play a role in alternative polyadenylation and mRNA shortening. In 3' UTR of fragile X homolog 1 FXR1 mRNA, the rG4 promotes alternative polyadenylation sites 3 times higher than canonical polyadenylation site to produce shorter mRNA. In the absence of rG4 region in the construct, the canonical poly A site is favored to produce longer mRNA isoform. Beyond gene expression, rG force can act as structural scaffolds to assemble biomolecular condensates, particularly the stress granules, SG. Under stress conditions, the formation of protein RNA complexes temporarily store the untranslated mRNAs in stress granules, protecting them from degradation while helping the cell conserve the materials and energy for post-stress recovery, which is essential for cell survival during stress. In neuronal cells under oxidative stress, DNA, PTP6 protein binds rG4 RNAs with high affinity to coordinate the assembly of stress granules, which promote cell survival and reduce neuronal synaptic dysfunction and cell death. Without these rG force, the assembly of prosurvival complexes is missing, resulting in synaptic dysfunction. In osteosarcoma U2OS cells under stress, the RNA-binding protein G3BP1 interacts with rG4 containing RNAs to form stress granules, which protect cells from functional collapse. Conversely, BLM protein in stress granules can unwind the rG force with its helicase activity, thereby negatively regulating stress granules. In this paper published in Cell, RNA G-quadruplex forms scaffolds for alpha-synuclein aggregation. Purified alpha-synuclein binds RNA G-quadruplexes directly, leading to phase separation, assembly, and sol-to-gel phase transition. In neurons, rG4 in synaptic mRNAs can co-aggregate with alpha-synuclein, eliciting synaptic dysfunction. The drug that prevents RNA G-quadruplex phase separation can attenuate alpha-synuclein aggregation, neurodegeneration, and progressive motor deficits in mice. rG4-triggered neurodegenerative synucleinopathies can include Parkinson's disease, dementia, and multiple system atrophy. rG4 functions are not limited to mRNAs. rG4s are widely found in non-coding RNAs including microRNAs, tRNA-derived small RNAs, piRNAs, and long non-coding RNAs in microRNA maturation, tRNA regulation, piRNA regulation, and lncRNA functions such as TERA in telomere stability. In long non-coding RNA, lncRNAs, rG force can recruit regulatory proteins to regulate downstream gene expression. For example, rG4 in lncRNA, reg1CP can actively recruit helicase FANCJ. Once recruited, FANCJ catalyzes the unwinding of double-stranded DNA structures that normally represses downstream transcription from transcription start site, TSS. This unwinding effectively lifts the blockade on transcription, leading to increased expression of downstream protein-coding rG4 gene. Consequently, the upregulation of rG4 mRNA and protein promotes cancer cell cycle progression and oncogenicity. Another mechanism of rG4 regulatory function is by decoying protein factors. For example, the rG4 in lncRNA G-quadruplex, which is upregulated in colorectal cancer, CRC, binds DHX36 helicase to competitively decoy the helicase away from its real targets. Normally, the RNA helicase DHX36 targets thousands of mRNAs, unwinds their rG4s, allow the ribosome to smoothly traverse the mRNA, and increase the mRNA translation. With DHX36 being decoyed away and sequestered by lncRNA G-quadruplex overexpressed in colorectal cancer cells, or by DHX36 knockdown, the protein translation for these mRNAs is stalled. Instead, the stalled mRNAs are accumulated in stress granules and linked to cellular stress responses that ultimately promote cancer cell survival, proliferation, and metastasis. Post-transcriptional modification of RNA, particularly M6A on mRNAs, is a layer of gene regulation studied as new research field of epitranscriptomics. M6A modification by methyltransferases metal-3 and metal-14 occurs preferentially near rG4 regions. First, metal-14 recognizes and binds to rG4, associated regions containing DRACH motif sequences, and then activates the catalytic activity of metal-3 to complete the addition of the M6A modification. That is, the rG4s influence the efficiency and location of M6A addition, thereby creating a specialized microenvironment that favors epitranscriptomic outcomes. This sequential mechanism ensures that M6A modifications are precisely targeted to regions adjacent to rG4s. Such precise localization is believed to be a key determinant of the biological outcome, whether an mRNA is destined for rapid degradation or translation regulation. The interplay of rG4 and M6A in translation regulation is exemplified in VEGF translation regulation. Here, rG4 guides the M6A modification in the internal ribosome entry site IRES, in the 5' UTR of VEGF mRNA. The M6A modification at the location has dual roles. 1. It blocks off the upstream open reading frame, UORF, translation, which effectively prevents abnormal or premature translation that may compete with main MORF translation. This resets the translation start site to the main MORF for VEGF protein production. 2. Its M6A reader protein YTHDC2 recruits translation initiation complex, including EIF4G, to start VEGF translation from the main MORF. By this mechanism, the presence of M6A ensures that the translational machinery is directed preferentially toward the MORF, thereby increasing VEGF protein output. This regulatory outcome is a result of a coordinated interplay between rG4 structure and epitranscriptomic marking. This finely tuned interplay is particularly crucial for VEGF required in rapid angiogenesis in solid tumor progression. The aberrant rG4 molecular and cellular functions are associated with many diseases. For example, rG4 staining in brain hippocampal regions increase linearly with age from young to old. rG4s are involved in neurodegenerative diseases such as Alzheimer's disease, Parkinson's disease, dementia, and multiple system atrophy. In Alzheimer's disease, chronic rG4 can cause protein aggregation that leads to neurodegeneration. rG4 are pathologically involved in many cancers such as glioblastoma, breast cancer, prostate cancer, and pancreatic cancer, in all cancer hallmarks of evading growth suppressors, sustaining proliferation, activating metastasis, telomere motilization, angiogenesis, and resisting cell death. Many key oncogene and tumor suppressor mRNAs have rG4 regulatory elements. These rG4s can well be therapeutic targets of rG4 ligand class. In cardiovascular system, the master cardiogenic transcription factor NKX25 mRNA is post-transcriptionally regulated by rG4 in its 5' and 3' UTRs. Disruption of the rG4 regulation can cause defects in heart development and congenital heart defects. In metabolic system, rG4 regulates the biogenesis of obesity-related microRNA 26A1, which alters AKT signal transduction pathway, insulin sensitivity, and lipid biosynthesis. In repeat extension disorders such as Fragile X syndrome, the CGG repeat-derived RNA G-quadruplexes, CGG G4 RNA, is pathogenic, causing binding protein aggregation, tremor, and ataxia syndrome. In viral infectious diseases such as Epstein-Barr virus, encephalitis, SARS, COVID, HIV, Zika, West Nile, and herpes viruses, the viral rG4s regulate viral infection, life cycles, and immune evasion. To systematically study rG4 profiling is essential. Arraystar rG4 microarray is a powerful technology for rG4 profiling. In the procedure, rG4s are not directly immunoprecipitated for detection due to the dynamic nature of rG4 in vivo. Instead, the landscape of rG4 in the cells is first countermarked by dimethyl sulfate (DMS) treatment. Unpaired A, C, G bases in an unfolded RNA are preferentially modified by methylation, whereas the paired bases within rG4 are protected from modification. The DMS-treated RNAs are completely denatured and refolded in the presence of potassium ions. The denatured unmodified rG4s can refold back to rG4 structure, whereas denatured modified non-rG4 sequences cannot. The total RNA is then extracted from the cells and immunoprecipitated by rG4 antibody, bG4. The immunoprecipitated RNA fragments containing rG4s are demethylated for reverse transcription, synthesized for fluorescently labeled cRNA, and detected on the rG4 microarray. There are significant advantages by this rG4 profiling technology. DMS is highly permeable to the cells and modifies unfolded RNA sequences efficiently in vivo within the cells. The methylation is reversible by demethylation without interfering with later enzymatic reactions. rG4 microarray is sensitive at rG4 detection and quantification, particularly good for rG4s that are dynamic and low in abundance. The sensitivity and accuracy are far better than sequencing. In a brief comparison with current DMS rG4-seq for rG4 profiling, this method similarly uses DMS to treat cultured cells to countermark the folded rG4 structures. The RNA is then purified and refolded in solution. Reverse transcription stop library is constructed, where rG4s cause reverse transcriptase to stall, that is, RT stops, and the stops are identified by sequencing. In the control reaction, lithium ions are used to replace potassium ions, in which rG4s cannot refold. Here, the rG4s are identified by reverse transcription stops, not by rG4 antibody immunoprecipitation. The limitations of this method are DMS methylation of M1A and M3C not related to rG4, which can also cause RT stops and increase the likelihood of false positives. Also, sequencing is not as sensitive or accurate as microarray for rG4s at low abundance. In comparison with another rG4 profiling method, UV-RIP-seq. This method uses UV cross-linking to fix rG4 binding protein to the rG4. The rG4s are immunoprecipitated by antibody against the rG4 binding protein, or alternatively by antibody against the rG4 structure, known as RNA immunoprecipitation, RIP. The RIP'd RNAs are sequenced and analyzed. However, the UV cross-linking may produce false positives due to RNA-RNA cross-linking. When UV cross-links the binding protein with rG4, immunoprecipitation with rG4 antibody may be blocked, causing false negative. Again, this method uses sequencing for detection, which is less sensitive and less accurate when transient dynamic rG4s are in low abundance. For these reasons, Arraystar rG4 microarray is recommended as much better rG4 profiling technology. Arraystar rG4 microarray data are provided with annotation and analysis. For example, the rG4 host genes, rG4 sequences, and rG4 location in mRNA regions, 5' UTR, CDS, or 3' UTR. Graphical analyses and visualization, such as scatterplot, volcano plot, hierarchical clustering heat map, pathway analysis, gene ontology analysis are provided to gain the insight into rG4 biology. On human rG4 array, close to 14,000 rG4s are probed. These rG4s are comprehensively collected from rG4 databases and scientific publications. To use rG4 microarray profiling, the cells or tissues, for example, disease versus healthy control, are treated with DMS in vivo. The total RNAs are extracted, immunoprecipitated, and used for rG4 microarray profiling experiment. The differentially expressed rG4s are identified and can be validated by rG4 qPCR. As an rG4 research roadmap ahead, the differentially expressed rG4s can be studied not only by their host RNAs where they reside, but also by identifying the rG4 interacting proteins and their functions, for example, by mass spectrometry. rG4 binding protein knockout, knockdown, or overexpression can provide valuable information. Small molecule rG4 ligands are available to stabilize, for example, pyridostatin, PDS, or destabilize, for example, PHPC, rG4 structure, and observe the consequences. rG4 mutation, for example, changing G to A, or rG4 expression perturbation by antisense oligo ASO, or by siRNA have been used to study rG4 effects. rG4 on mRNA translation can be studied by reporter assays. Biomolecule assembly or condensates can be studied by hybridization proximity mass spectrometry, HyPro-MS, fluorescent in situ hybridization, FISH, surface plasmon resonance, SPR, or fluorescence resonance energy transfer, FRET, rG4 and M6A modification by miRIP qPCR. rG4 can be predicted by computational analyses and AI machine learning. Common rG4 biochemical analyses include RNA immunoprecipitation, RIP, by BG4 antibody, chemical mapping by DMS or N-acetyl-imidazole, NAI, rG4 ligand binding or staining, for example, by Biotask, rG4 reverse transcriptase stopping, lithium-ion-based primer extension using DMS-LIPE, or using selective 2'-hydroxyl acylation, SHA-LIPE, for rG4 mapping. Circular dichroism, melting temperature, and NMR are some biophysical methods to analyze rG4 structures. I hope the information so far is helpful for your rG4 research. Thank you for your interest. Please visit us at Arraystar.com.